particularly a great pleasure for me to introduce the next speaker, Steve Vucic. So Steve actually initially contacted me. He was a neuromuscular fellow at Massachusetts General Hospital in Boston, and he said he wanted to come back to Australia and do some research. So we sat down, had a chat, and he had no support at that stage, but the MND Research Institute of Australia introduced a clinical fellowship, and Steve was the lucky recipient, and that supported him through his PhD studies. And really, Steve started looking at the mechanisms of motor neurone disease. We've heard about the genes. There must be more than just genes to trigger the disease. And in fact, there was a lot of controversy at the time whether this was a neurodegenerative central disease problem, because many people regarded it still as a neuromuscular condition, and it was looked after by physicians who did EMG, but they didn't have any understanding that there was clear brain involvement, and as Ian has mentioned, cognitive changes, and these things have really come to the fore. So Steve was right at the cutting edge of understanding cortical hyperexcitability, and the techniques that he developed in his PhD have gone on now, and they've been commercialized by a company in the United Kingdom, MagStim, and hopefully they'll be distributed worldwide. The other thing that it's been great for Steve, he's developed his own research team. He's gone from PhD student, senior lecturer, associate professor, professor of medicine at Westmead Hospital in St. Joseph's. He started up his own clinical service there. It's one of the largest clinics in Australia, if not the largest. So he's working as a clinician and a researcher. He's built his own team up. And we heard David Gillespie mention the Medicines Research Future Fund, and Steve has been able to now tap into that and is on the cusp of announcing a very large-scale clinical trial that will be driving some of the things that he's going to speak about. The other thing to mention is he's also the data center for the Neals Consortium in the United States. So they've got a clinical trial going on now, 14 US sites. All of the data is being sent to Steve as the data center using his techniques and analysis. So it's great that Steve is here, and he's going to give us a presentation from his research looking at ALS in the new century, and particularly towards therapeutic strategies.
Well, thank you, Matthew, for that very kind introduction. And none of this work would have been possible with Matthew's help, guidance and mentorship, and also without the great generous support of the MND Research Institute of Australia and the MND Association of Australia and New South Wales. Look, I'd like to start off from the beginning, and to my surprise, motor neuron disease was not described by Charcot, but rather by this gentleman, Sir Charles Bell, who of all things was a surgeon. But he was a remarkable man. He was credited at differentiating between the motor and the nervous system. And so he was the first man who described motor neuron disease. But it was really Charcot, some 150 years before genetics, or 100 years before even genes were known, who really nailed the pathophysiology of ALS, or rather motor neuron disease as it was called then, where he identified that there was abnormalities in the brain. And it was Charcot who first suggested that perhaps this is more than just a disorder of the anterior horn cell, and he coined the term amyotrophic lateral sclerosis to describe the pathology both in the motor neurons but also in the central nervous system. And this is what Charcot would have been seeing, progressive motor syndrome affecting the arms and the legs and the bulbar region, the throat and the swallow, with brisk reflexes, which was inexorably progressive. His contemporary David Gowes saw the same thing, but he looked at it in a different way. He proposed that motor neuron disease was really just a disorder of the motor neurons, and that the upper motor neurons of the central nervous system was a byproduct. And unfortunately, Gowes' view was held for over a century until our close collaborator and friend Andrew Ison in the early 90s came up with the dying forward hypothesis. So Andy Ison turned the motor neuron disease world on its head, and he said, well, he felt that motor neuron disease was, in essence, a dementia, a primary disorder of the central nervous system, and this was based on a number of important but poignant clinical observations, one of which is this is a uniquely human disease with very poor reproducibility in the animal models, and plus other observations. naturally to say he was argued against quite vehemently. And in this sort of hotbed, this is just the typical clinical features of ALS include both upper and lower motor neuron dysfunction, brisk reflexes, progressive muscle wasting and weakness, etc. But in this hotbed of argument as to whether motor neuron disease is purely an anterior horn cell disorder, that is, whether it just starts at a spinal cord level or whether this is something higher, this is where I arrived. And as Matthew mentioned, I'd come back from Mass General, had no funding, and actually went to see Matthew to do some peripheral nerve excitability work on autoimmune neuropathies, and he said, this is far more important and interesting. And I haven't, his insight was quite ingenious at that stage, and I've enjoyed every moment of it, of the research. But that's me in 2005 as the inaugural MND New South Wales Clinical Research Fellow, which was quite instrumental to the shaping of my ensuing career. So I had a lot of hair and a lot of weight back then, as you can see. All the hair's gone. And so in that setting, we went about to resolve whether motor neuron disease starts centrally or peripherally and whether there's any link. The first step was to develop a reliable method of assessing brain function, and one of those was a threshold tracking transcranial magnetic stimulation, whereby we stimulate the motor cortex relatively painlessly using a magnetic coil, and in real time we actually measure the output. We measure the response from the arm or the leg, and by determining the current required to produce a response, we can tell whether the brain is hyperexcitable or not. And in actual fact, Justin was one of my early guinea pigs. He never spoke to me after that. I'm just kidding. We've had a good relationship since then. And so one of the very important biomarkers of brain function is this thing called short interval intracortical inhibition. And we now know that this is mediated by these intracortical interneurons that are very important for thinking and all sorts of things. And they act by a chemical called GABA. And we know that. And so we wanted to apply this in both sporadic and familial ALS cohorts to see whether indeed there was brain dysfunction. So this is, I might add, this was about 8 or 9 years before, or some years before the discovery of the C9orf gene. What has also emerged is that ALS is quite a markedly complex pathophysiological process. It is more than just a monogenetic disorder. There are a whole range of interconnecting molecular abnormalities that interact with one another, and one of which is glutamate excitotoxicity, which is highlighted here. And through excessive stimulation of the motor neuron or the motor neurons, there is degeneration. This is one of the mechanisms by which we feel that ALS propagates. And so in that setting, we wanted to focus on that, but in a clinical setting, because we'd known for some time that at a molecular level, there was certainly good evidence both in the mouse model and also in molecular studies in humans that there were abnormalities of glutamate metabolism. There was too much glutamate around. The way that glutamate was handled by the body was impaired, and there was a predisposition for the motor neurons to express the so-called glutamate receptors, which made them more vulnerable to damage. And so what did we find using our threshold tracking technique? So the first study we published in Brain, which was actually my first brain paper I ever published. I was very, very happy with that. Brain is one of these sort of icons that we all look up to in neurology, and in actual fact, David Gowes was one of the editors of Brain way back in the 19th century. And so what we discovered was that indeed cortical hyperexcitability was evident in ALS, as indicated by a marked reduction in this intracortical inhibition. Moreover, this was more prominent in patients who had disease in their arms or limb onset disease, because that's where we recorded the response. And it was more prominent in those patients who had a relative preservation of their lower motor neuron function. So these are the people that are in black, in the black bar graph. In addition, there was a strong link between central or hyperexcitability and lower motor neuron dysfunction as measured by these standardized markers of peripheral nerve loss. And this was really the first study to show this link, suggesting that perhaps the dying forward hypothesis was correct. It wasn't conclusive, but it was suggestive. Some years later, jumping way forward to 2016, Dr. Menon in my lab, who is the current Beryl Bailey Fellow, Matthew and I are her supervisors, wanted to look at the very earliest effects in the disease process, and so she recruited patients very early on who had relative preservation of their lower motor neuron function. In actual fact, from the target muscle, there was no lower motor neuron dysfunction, using a whole range of techniques. And indeed, she found there was marked hyperexcitability, which preceded the lower motor neuron loss by up to 12 months. And this was really one of the very important papers that proved the central theory of ALS. We next observed that there is dissociative hand loss. This has been known for some time. That is, the muscles outlined in red are predominantly affected in ALS compared to the what we call the hypothenar muscles, so that is the muscles that are just over here. And it was postulated that perhaps this had a cortical origin because these muscles had a greater cortical representation. And indeed, we looked at this and found this was the case. Whilst hyperexcitability was evident across the three target muscles, the abductor pulsus brevis, first dorsal interosseous, and ADN that we studied, it was more prominent in those affected muscles, suggesting that perhaps this hyperexcitability is driving the process. Now, one other thing that we observed, and again, Dr. Menon and myself and Professor Kiernan observed this so-called split hand plus sign. That is, when the patient tries to abduct their thumb, the distal part of their thumb bends. That's despite the fact that the same motor neurons, the CAT1 and the same nerve, innervates the same muscle. There was quite marked wasting of the abductor pulsus brevis muscle. And so we wanted to see, well, does cortical hyperexcitability play a role in this? Because we knew that the input to the flexor pulsus longus from the brain is somewhat different to the abductor pulsus brevis. And again, we wanted to confirm the first step, whether this was indeed a specific feature for ALS, and indeed it was. It was not seen in any of the, am I not loud enough? It was not seen in any of the neuromuscular disorders that look like ALS. I might speak softer. And again, indeed, this was the case. There was a quite marked increase in hyperexcitability, which was more prominent over the targeting muscle. So during this course, during these years, we kind of felt that indeed hyperexcitability was gaining important reputation as a pathophysiological mechanism in ALS. Okay. Sometime later, Robert Henderson's group, working with Matthew Kiernan and his student Matthew Devine, looked at how ALS spreads, because this is critical for the understanding of what's driving the disease. And they came out with this quite important paper looking at different patterns of spread, and they described both what they termed vertical and horizontal spread, but suggested that the corticomotor neurons from a clinical aspect were very important for this spread. And indeed, we wanted to look at this with Parvati Menon and Dr. Givasinga in my lab. So we looked at a large cohort of patients, looked at their spread, and then measured brain activity. And indeed, we found that there was an asymmetry in hyperexcitability. It seemed to start in one area of the brain, usually the dominant side, and then it spread to the other side, and it was more prominent in the cortex that was opposite to the spreading to the affected limb, because as you all know, the brain is upside down, the right innervates the left, and so on. And indeed, when we looked at patients who had horizontal and vertical spread, again, there was asymmetry in hyperexcitability, such that this cortical hyperexcitability seemed to be driving the mechanism of disease spread. One last thing, with a Japanese colleague, we looked at, well, does hyperexcitability matter in terms of survival, and is it an adverse prognostic factor? And indeed it was. So those ALS patients that had a marked increase in hyperexcitability, did more poorly than those that had less level of hyperexcitability. So this was an adverse prognostic factor. Now, changing tack slightly from sporadic MND, where we were fairly convinced that the corticomotor neurons are important, we looked at familial motor neuron disease, and we looked at three mutations: the SOD1, the FUS, and the more recent, the C9orf. And in collaboration with Professor Garth Nicholson, we looked at three large Australian families and identified that hyperexcitability was a feature in those patients that were clinically affected, suggesting a similar pathophysiology to sporadic ALS. What was interesting was that the carriers of the mutation did not, were clinically normal, but they were also cortically normal. Their level of brain excitability was no different to a healthy control. However, in a subset of these patients, hyperexcitability developed on average 6 months before the onset of lower motor neuron disease. And again, what this suggests is that being born with the gene is a predisposing, not a causative factor. And this is one of the discussions I've had with Garth. And there are other and additional factors that are very important in triggering the disease within patients. In FUS mutations, again, hyperexcitability is a feature. And also more recently, Nimish and Givasinga looked at the C9orf patients and identified hyperexcitability as a feature in the clinically affected patients. But in those that were mutation carriers, the level of brain excitability was normal. Again, showing that there is a pattern here that irrespective of whether you're sporadic or genetic, which obviously are perhaps sporadic ALS are probably polygenetic plus other things, cortical hyperexcitability is an important mechanism. It is perhaps the driver of both disease onset and one of the mediators of disease spread. But motor neuron disease is not a uniform phenotype. It is, there are whole different ways that it can present, and one of the ways that it can present is this flail arm variant, or the man in the barrel, or as the Americans coined it, BAD, bibrachial amyotrophic diplegia. But for a long time, this was not thought to be a form of motor neuron disease because it didn't have a lot of clinical upper motor neuron signs. It remained confined to the arms for a prolonged period of time, and it did not have the same type of course. But what in studies that we conducted in 2007, we identified there was subclinical upper motor neuron dysfunction. Hyperexcitability was a marked feature in this flail arm variant and was more prominent even than the sporadic ALS patients. And more recently, with work that we've done with Glenda Halliday and her students, one of the flail arm variants has shown widespread TDP-43 deposits in the brain. So there you go. One other variant is the flail leg variant, which is a form of motor neuron disease that starts in the legs and remains localized there for a prolonged period of time. And again, these patients have hyperexcitability, but if the hyperexcitability is prominent, they have a more adverse prognosis than those who have a relative preservation of this. Now, it was posed to us for a long time whether or not this hyperexcitability was a simple cortical plasticity phenomena. In actual fact, one of the molecular genetics groups from Italy who were focused on animal models proposed that hyperexcitability was neuroprotective. And so we looked at this question in a large cohort of ALS patients and what we call mimicking disorders, people that look like ALS but are not, and identified quite marked differences between ALS and non-ALS. So hyperexcitability is prominent in ALS but not in the non-ALS group, suggesting that this is not a simple cortical plasticity phenomenon. But this also led to a different spin-off, and that being of a diagnostic test. We found that if we applied this as an objective diagnostic biomarker, you can reliably differentiate ALS from non-ALS patients in a clinical setting. And all you needed to do is to test 2 patients in order to make the diagnosis of ALS, which is crucial because an early diagnosis is absolutely critical, I feel, in future ALS trials to have a more therapeutic compounds. And based on these studies, as Matthew has mentioned, this technology has been commercialized with industry partner Magstim under the name MagExcite, and we are hoping to roll this out early next year to be both a diagnostic but also prognostic biomarker in ALS. Have I got some more time? Now, changing tack slightly, how does this translate to therapeutics? Well, up until recently, the only effective therapy, albeit partially effective therapy that we've had for ALS was riluzole. This agent works by blocking the release of glutamate, and as such is an anti-glutaminergic agent. However, it prolongs life by 3 to 4 months, and we thought, well, how does this work? At a systems human neurophysiological level, what riluzole does is it partially normalizes hyperexcitability, increases SICI from here up to here. And in a more recent longitudinal study conducted by Nimish Givasinga from my lab, who's also been funded by the MND RIA, we found that when we longitudinally followed patients with riluzole, there was a wearing off effect. It peaked at 8 weeks, but by 3 months, the effect was lost. And so understanding what underlies this loss of effect would be critical to trying to enhance the efficacy of riluzole. Now, more recently, Brian Wenger and colleagues from Mass General have looked at this in another way, the issue of hyperexcitability in a cell model using induced pluripotent stem cells derived from ALS patients, and they again identified hyperexcitability at a cellular level. And when they blocked this hyperexcitability with a drug called reticabine, which is a simple anti-epileptic that we have around, they found it to be neuroprotective, at least in a cell model. Based on these studies, the Neals Consortium has funded a large phase 2 study, which is currently ongoing, looking at the effect of this drug in ALS. And Matthew and I have been invited to be the international data center for this trial, and hopefully we'll have the results by mid-2018. And so this leads us to take one step back, and I think ideas are quite important because these form future research, and simple ideas can lead to big discoveries. So one of the issues, whether indeed ALS is a dementia with this so-called river of hyperexcitability emanating from the motor cortex driving the spread. And one of the spin-offs of this has been in stem cell therapies, whereby current stem cell approaches which dictate injection of neuronal stem cells at a spinal cord level have been largely unsuccessful, albeit safe. And so one of the authors in this paper, Stuart Rutkover, has suggested that perhaps we need to target the upper motor neuron system as well. In the last 2 minutes that I have, I just want to move away slightly from hyperexcitability to talk about neuroinflammation, which is another thing that the MND RIA has funded with my colleagues Fiona McKay and David Booth at Westmead Hospital. It has been known for some time that neuroinflammation can regulate the rate of disease progression. There's the good and there's the bad neuroimmune system. So the good is highlighted here. So this is the regulatory T cell monocyte type 2 system, which seems to be predominant in the early stages of ALS. And so during this course, the rate of disease progression is slower. And then at some point in time, and for some unknown reason, there's a switch to the cytotoxic or the neurotoxic response, and that's when the disease accelerates. Stanley Appel and his team in Texas have shown that these regulatory T cells correlate with a slower rate of disease progression. So the higher your Tregs, the lower the rate of disease progression. And so we wanted to focus on this to see whether we can keep ALS patients perpetually in a Treg state. And initially, Fiona, in collaboration with Bradley Turner at the Florey, we looked at initially human studies where we identified that indeed regulatory T cells, which were extremely well phenotyped, far better than previously, did indeed correlate with a rate of disease progression. So the higher your regulatory T cells, the slower your rate of disease progression. And then we then upregulated these regulatory T cells in a SOD1 model of ALS using a particular compound called interleukin 2C. And what we found was quite astounding was that it significantly slowed down disease progression such that there was a marked prolongation of life from about 148 days to about 180 odd days in the animal model, which is a lot for a rat. That's what Bradley tells me. And this is under consideration. Well, it's been provisionally accepted by JAMA Neurology. But this research is quite important because it has fostered or generated interest in a large trial that is going to be announced very shortly, looking at an agent that predominantly acts on the regulatory T cells. This will be hopefully the largest phase 2 trial in Australia, led by myself and Matthew from Sydney University, and incorporating 6 centres across Australia to be a truly national study from east to west coast. We will update you very short on that. But in the last minute, I'd like to leave you with this thought. ALS is a very complicated disease. And this is a great review written by the cognoscenti of ALS thought, Matthew and Andrew Ison and Michael Swash and Hiroshi Mitsumoto, where they propose that perhaps you were born to develop ALS. It's a in utero exposure that leads you to develop ALS, whether that be a genetic predisposition or whatever. And then during one's life, you meet all of these epigenetic factors, such as environment, oxidative stress, glutamate, excitotoxicity, etc. Amar Al-Chalabi and colleagues from the UK have suggested that on mathematical modelling, you need 6 different factors to be exposed to in a sequential order to get ALS. But clearly what we need to understand is that ALS has a long prodrome, that's what we feel, and that we get the patients way down here when it may be too late for neuroprotective therapies. We need to get the patients down here. So combining clinical, physiological, molecular, genetic biomarkers to diagnose patients very early is crucial and remains a point of investigation. So I'd like to stop there, and there's a lot of people I'd like to thank. First of all, the MND RIA for their indelible and wonderful support. Without MND RIA support, none of my research would be possible, which has led to a number of other grants, including a number of NHMRC grants. I'd like to thank, obviously, Professor Matthew Kinn for his mentorship and support, and my team at Westmead, Julie and Linda, and all my research colleagues there, Garth Nicholson and Michelle Farah from Prince of Wales. Thank you.